Dear colleagues, I want to present you research titled as Posthypertonic Lysis of Erythrocytes when using Amphiphiles in varying temperature. Currently, for the rescue of wounded and injured defenders and civilians of Ukraine, the creation of reserves of donor blood and its components to provide hospitals with cryopreserved blood components have a crucial importance. The use of cryopreserved blood components in hospitals and medical centers is extremely important due to reparative processes in the healing of wounds and burns, injuries, diseases, and other serious pathologies. Unfortunately, the stocks of human donor blood in Ukraine are significantly limited by a reduced number of donors, spread of bloodborne diseases, and increased demand for blood due to the emergency and war. Storage of erythrocytes at total temperature causes the biological activity of cells, which enables them to be preserved for a long time. To minimize freezing damage, cryoprotective additives are pivotal. The permeating additive glycerol has been used to freeze erythrocytes in the early 50s. And currently, glycerol is the most commonly used erythrocytes cryoprotective agent. During thawing of cryopreserved erythrocytes, they are affected by cryodamage factors, which include changes in the concentration of salt and cryoprotective agent e.g. glycerol. To assess the effect of these factors on cells, posthypertonic shock model is used, in which cells after incubation in dehydration medium are transferred to the rehydration one. Previously, there was demonstrated the ability of amphiphilic compound to reduce posthypertonic lysis level of human erythrocytes. The research aim was to study the effect of the temperature on human erythrocytes preserved after combined action of posthypertonic shock in Amphiphiles. Posthypertonic shock was initiated by transferring erythrocytes from dehydration medium into rehydration one. The level of erythrocyte hemolysis was determined by spectrophotometry. Amphiphilic compounds belonging to the different classes of surfactants, which are characterized by different transmembrane distribution, were used in this research. Amphiphilic compounds were added into rehydration medium prior to transfer the cells based on the results of this research. Incorporating positive charged trifluoroperazine into the immunomonoline causing stomatocytosis. Incorporating both anionic sodium dicyl sulfate and non-ionic dicyl pronoside mainly into the outer monoline causing echinocytosis. To evaluate and compare the efficiency of amphiphilic compounds under pHs condition of erythrocytes, we used the notion of plateau, values of effective concentrations, and maximum antihemolytic activity. The plateau of a amphiphilic compound is a range concentration within the minimum level of erythrocytes hemolysis observed. Effective concentration of compound is the one corresponding to the midpoint of this plateau. The maximum antihemolytic activity was calculated by this formula. From this slide, it can be seen that at 0 Celsius degree, amphiphilic compound decrease posthypertonic hemolysis of erythrocytes. The presented dependencies are characterized of three areas. First, a decrease hemolysis of erythrocytes is observed, and then the plateau of effective concentration is formed. A further increased concentration of amphiphils leads to the cell damage. It can be noted that the plateau concentrations are different, so trifluoroperazine has a narrow plateau, while dicyl-like pronoside has the widest. From obtained dependencies at 0 Celsius degree, were determined values of effective concentration and calculated values of antihemolytic activity, which are presented in Table 1. In spite of 0 Celsius degree at 37 Celsius degree, all amphiphilic compounds does not reduce the level of erythrocyte hemolysis. It can be seen that all amphiphilic compounds show sufficiently high antihemolytic activity at 0 Celsius degree. Therefore, it was interesting to test how the found protective effect of compounds in the conditions post-hypertonic shock is stable, and to assess the state of cells, they were exposed to posthypertonic shock and amphiphiles at 0 Celsius degree. Then they were heated and kept for 30 minutes at certain temperature. The data presented in this slide. Can be seen that the level of hemolysis of control cells does not depend on temperature changes. When using trifluoroperazine, the development of erythrocyte hemolysis is observed at 20 Celsius degree and higher. In case of dicyl glycoperoxide, just a temperature of 37 Celsius degree. And when applied sodium dicyl sulfate, the level of cell hemolysis does not depend on temperature changes. Thus, it was established that erythrocytes which were preserved after posthypertonic shock action in the presence of trifluoroperazine and dicyl glycoperoxide are sensitive to temperature increase, but in presence of sodium dicyl sulfate, the cells are resistant when temperature increasement. So, at further experiments, we using dicyl sodium sulfate. Erythrocytes are characterized by transmembrane distribution of phospholipids. In particular, phosphatidylserine is a marker phospholipid of the inner monolayer of the lipid bilayer. Irreversible damage to the erythrocytes may be indicated by change in the transmembrane distribution of this phospholipid, via its exposure on the outer surface of the cell, which can be determined using annexing. Since the cells after the effect of posthypertonic shock showed resistance for further heating using sodium dicyl sulfate salt, it was expedient to study the transmembrane distribution of phosphatidylserine, namely to determine the number of annexin-labeled erythrocytes by flow cytometry. The amphiphilic compound sodium dicyl sulfate was used in two concentrations. First, the effective concentration at which the maximum antihemolytic activity was observed is 400 micromole per liter. The concentration of compound at which antihemolytic activity is approximately 45% is 180 micromole per liter. The experiment was performed in two versions. In the first one, human erythrocytes were incubated for 20 minutes in a temperature of 0 Celsius degree in a physiological solution. Control was cells incubated in physiological solution in the absence of sodium dicyl sulfate. In the second variation, human erythrocytes were exposed to posthypertonic shock at 0 Celsius degree, while the amphiphilic compound was present in the rehydration medium before introducing cells into it. The asymmetry of erythrocyte membrane was assessed by means of flow cytometry using the annexin 5-FITS apoptosis detection kit. The results of studies of human erythrocytes by flow cytometry show that the content of control erythrocytes in a set of amphiphile that bound annexin 5 was 0.34%. In the case of using sodium dicyl sulfate in a physiological solution, the number of positively labeled cells largely depends on the concentration of amphiphilic compounds. If sodium dicyl sulfate at low concentration 180 micromole per liter causes the appearance of almost 5% of annexin-labeled cells, then at a high concentration 400 micromole per liter is about 23. In the case of conducting erythrocytes posthypertonic shock at 0 Celsius degree with the addition of sodium dicyl sulfate in a low concentration, the percentage of positively labeled cells was slightly higher, almost 6%, while the use of high concentration of sodium dicyl sulfate increased the number of annexin-labeled cells, almost 38%, compared to corresponding indices in a physiological solution. Therefore, with the increased concentration of sodium dicyl sulfate, the percentage of positively labeled cells increases, which indicates the redistribution of phosphatidylserine from the inner to the outer monolayer of the lipid bilayer of human erythrocyte membrane. It should be noted that when using a low concentration of sodium dicyl sulfate in both variants in the experiment, In physiological solution and under the posthypertonic shock conditions of erythrocytes, these indexes differ slightly. The results obtained using the method of flow cytometry indicate that dependence of the transbiliary distribution of phosphatidylserine in erythrocytes membrane on the concentration of the amphiphilic compound, sodium dicyl sulfate. So therefore it was possible to choose such a concentration of the amphiphilic compound at which it would be able to show significant antihemolytic activity and not affect the transmembrane redistribution of phosphatidylserine in the membrane. We truly appreciate support of the Society for Cryobiology and grateful for the opportunity to participate. Thank you for attention.